Welcome back to Anatomiqa. In the last session, we completed the histology of cardiac muscle, which includes the specific features, structure of intercalated disc and the dyad, the features which could be identified in the photographs of the histological sections of cardiac muscle. In this session, we will discuss on the features and structure of smooth muscle fibers. Smooth muscle fibers are elongated, spindle-shaped with a single nucleus present in the bulged central region. The ends are tapered or narrowed. The fibers are arranged in fascicles. Smooth muscle is specialized for slow, steady contraction and is controlled by a variety of involuntary mechanisms. Fibers of smooth muscle are also called visceral muscle. To reinforce, they are elongated, spindle-shaped, tapering, and non-striated cells, each of which is enclosed by a thin basal lamina and a network of reticular fibers, the endomysium. The connective tissues serve to combine the forces generated by each smooth muscle fiber into a concerted action, for example, peristalsis in the intestines. Smooth muscle cells may range in length from 20 micrometer in small blood vessels to 500 micrometer in pregnant uterus. Each smooth muscle fiber or cell, which is spindle-shaped, has a single long nucleus located in the center of the cell's central broadest part. The cells strain uniformly along their lengths, and to achieve the tightest packing, the narrow part of one cell lies adjacent to the broad part of neighboring cells. Such an arrangement viewed in cross section shows varied size of cell diameters, with only the largest sections at the broad part containing a nucleus. All cells are linked by numerous gap junctions. Let us now discuss the ultrastructure of smooth muscle cells. The short membrane invaginations are often present on the cell surface of the smooth muscle called caveolae. Caveolae of smooth muscle cells contain several pumps and ion channels and may serve to organize protein signaling calcium release at myofibrils. Adjacent smooth muscle cells communicate through gap junctions. Concentrated near the nucleus are mitochondria, polyribosomes, rough endoplasmic reticulum, and the Golgi apparatus. The myofilaments of the smooth muscle are not regularly arranged in a group, like in skeletal muscle. Because of this, there are no myofibrils and sarcomeres and hence appear non-striated. T-tubules are absent in smooth muscles and the fibers have rudimentary sarcoplasmic reticulum. The thick and thin myofilaments in the smooth muscles are oriented obliquely, crisscrossing through the muscle cell. The thick and thin myofilaments are arranged at angles to each other as they run through the cell. The thick filaments are made of myosin and the filaments have a less regular arrangement among the thin filaments and fewer cross bridges than in striated muscle. Each thick filament is surrounded by 12 to 15 thin filaments. The myosin are not always present in the sarcoplasm. The myosin molecules aggregate into thick filaments in response to stimulus before the contraction begins. The thin filaments are composed of actin and tropomyosin and lack troponin. The actin filaments in smooth muscle run from one side of the cell to the other. A sarcomere-like unit is composed of the actin filaments, which are anchored to the dense bodies in the cytoplasm as well as dense plaques on the cell membrane. These dense areas are composed of the protein alpha-actinin found in the Z-lines of striated muscle to which actin filaments are known to be attached. A network of intermediate fibers run between the dense bodies, providing an internal framework for contractile proteins to work. A dense body is analogous to the Z discs of skeletal muscle, anchoring the thin filaments in position. Coming to the mechanism of contraction, the sliding filament mechanism of contraction however, is basically similar to that in striated muscle. The characteristic contractile activity of smooth muscle is generated by myofibrillar arrays of actin and myosin organized differently from those of striated muscles. Calcium ions are supplied primarily from the extracellular environment. Caveolae replacing the T-tubules in the sarcolemma represent locations of high density of calcium channels present to facilitate calcium entry. Sarcoplasmic reticulum is present in the fibers but is less developed than that observed in skeletal muscle. Because troponin is absent in smooth muscle cells, cross-bridge formation is regulated by protein calmodulin and not by troponin-tropomyosin complex. When a smooth muscle cell is stimulated, external calcium ions pass through open calcium channels, caveolae, in the sarcolemma, with additional calcium released by the sarcoplasmic reticulum. The calcium, as it enters the sarcoplasm, it binds to calmodulin to form calcium calmodulin complex. This calcium calmodulin complex then activates an enzyme called myosin light chain kinase. Myosin light chain kinase in turn activates the myosin heads by phosphorylating them, that is, converting ATP to ADP and phosphorus, with the phosphorus attaching to the myosin head. The heads can then attach to actin binding sites and pull on the thin filaments. When the thin filaments slide past the thick filaments, the force generated by myosin cross bridges attached to actin is transmitted through actin filaments to dense bodies and then through neighboring contractile units ultimately terminate on the dense plaques of the cell membrane. This arrangement causes the entire muscle fiber to contract in a manner whereby the ends are pulled toward the center, causing the midsection to bulge in a corkscrew motion and the whole muscle contracts. Relaxed smooth muscle cell possess a smooth cell membrane appearance, but upon contraction, the cell membrane becomes scalloped and large membrane blebs or eruptions form as a result of inwardly directed contractile forces that are applied at discrete points on the muscle membrane. These points are presumably the dense plaques on the cell membrane to which the actin filaments attach. It has been hypothesized that in order for a single cell to shorten in a corkscrew manner, the contractile proteins in smooth muscle are helically oriented within the muscle cell. This helical arrangement agrees with earlier speculation that the contractile apparatus in smooth muscle may be arranged at angles related to the long axis of the cell. Such an arrangement of contractile proteins could contribute to the slower shortening velocity and enhanced force generating ability of smooth muscle. Smooth muscle cells also have an elaborate array of 10 nanometer intermediate filaments, usually composed of desmin. The intermediate filaments and actin filaments both insert into cytoplasmic dense bodies and plasma lemma associated dense plaques. Dense bodies contain alpha actinin and are functionally similar to the Z discs of striated and cardiac muscles. The attachments of thin and intermediate filaments to the dense bodies helps transmit contractile force to adjacent smooth muscle cells and their surrounding network of reticular fibers. Let us see how the muscle fibers are organized or arranged in the viscera. Usually, in most of the hollow viscera, the muscle fibers are arranged in a circular or longitudinal manner in the wall. In transverse section of the hollow viscera, that is when the hollow viscera is cut across the wall, the longitudinal fibers run along the wall or cut transversely with varied diameters of the fibers. The nucleus is observed only in the fibers with largest diameter in the section. The circular fibers arranged around the wall are cut longitudinally along the long axis of the fibers, showing the whole length. Muscle cells are grouped together in fasciculi. Adjacent fasciculi are separated by thin connective tissue. Each fasciculus acts as a contractile unit. Within a fasciculus, muscle fibers run parallel to each other, tapering ends of neighboring muscle fibers overlap with each other. Based on the gap junctions, smooth muscle is classified into two types, single unit and multi-unit. Single unit smooth muscle is also called unitary smooth muscle or visceral muscle. This type of smooth muscle is the most common type observed in the human body. Smooth muscle fibers usually form sheets of tissue and function in a coordinated fashion due to the presence of gap junctions between the cells. Due to the gap junctions, they are electrically coupled. If one cell supplied by a nerve is stimulated, the whole bundle of fibers contracts as single unit. These fibers are present in the walls of hollow organs. Single unit smooth muscle produces slow, steady contractions that allow substances such as food in the digestive tract to move through the body. Multi-unit smooth muscle, the second type of smooth muscle, are less commonly seen. They are composed of cells that rarely possess gap junctions and thus are not electrically coupled. The adjacent cells are independent from each other and innervated individually by separate branches. As a result, contraction does not spread from one cell to the next, but is instead confined to the cell that was originally stimulated. This type of smooth muscle is observed in the large airways of lungs, in the arteries, the arrector pili muscles associated with air follicles, and the intrinsic eye muscles which regulate light entry and the lens shape. They produce asynchronous contraction. Depending on the functional requirements, the smooth muscle fibers are arranged in different ways. Cardiovascular system. Smooth muscle cells are present in all blood vessels in the tunica media, excluding capillaries. The fibers are arranged circularly around the wall. Smooth muscle contraction and dilation in blood vessels, called as vasoconstriction and vasodilation respectively, controls blood vessel diameter, thereby controlling the distribution of blood and determining blood pressure. Respiratory system. Smooth muscle layers are present in the walls of the bronchi and bronchioles, arranged circularly around the walls. They help to regulate airflow into the lungs. Gastrointestinal tract. The extensive layers of smooth muscle are present in muscularis mucosa and externa in the wall of GIT. The muscle fibers are arranged in two layers. Inner circular, where fibers are arranged circularly around the wall, and outer longitudinal, where fibers are arranged longitudinally parallel to the long axis of the gut. They help move food down the GI tract via peristalsis and eject bile into the digestive tract from gallbladder. Urinary system. Layers of smooth muscle are found in the muscular layer of walls of ureter and bladder. In the ureter, the fibers are arranged as inner longitudinal along the long axis of ureter and outer circular layers around the wall. In the bladder, the fibers are not distinctly separated. They help in movement of urine out of the body. Male reproductive tract. 3 layers of smooth muscles are present in the muscular layer of vas deferens. Inner and outer longitudinal and middle circular layers. They help move sperm through the system and also functions to cause ejection of glandular secretions from the prostate, seminal vesicle and bulbourethral glands. Female reproductive tract. The myometrium of the uterus mostly consists of smooth muscle. The fibers are directed in different directions and cannot be demarcated. The fibers are stimulated by oxytocin to contract during labor to aid in delivery of the fetus. Ophthalmic system. Ciliary muscle arranged radially, longitudinally and circularly contracts and relaxes to change the size and shape of the lens. In the iris, the fibers are arranged radially and circularly to form the dilator pupillae and sphincter pupillae, which regulates light entry into the eye. This changes the amount of light entering the eye. Integumentary system. Hair follicles in the skin are associated with arrector pili muscles arranged obliquely between the hair follicle and epidermis. The fibers contract to elevate hairs in response to changes in temperature. And sphincters. The muscle fibers are arranged circularly around the wall, which regulates the contraction and relaxation, as in closing and opening of the hollow tubes. as in bile duct or anal canal or urethra. Smooth muscle is not under voluntary control and its fibers lack neuromuscular junction. Contraction is regulated differently in the smooth muscle of the viscera, the respiratory airways, or large and small blood vessels. The control can involve autonomic nerves, a variety of hormones, and local physiological conditions such as the degree of stretch. Whether smooth muscle fibers contract as small groups or throughout the entire muscle to produce waves of contraction is determined largely by the degree of autonomic innervation and the density of the gap junctions. Both conditions vary considerably in different organs. Now let us see the features which has to be identified in the sections under microscope. This is the diagrammatic representation of the longitudinal section of smooth muscle. The features observed or to be identified are spindle-shaped muscle fibers, oval nucleus in the center of the cell, and endomysium surrounding the muscle cell. Overlapping of the narrow edges over the central part of adjacent muscle fibers can be observed. This is the photomicrograph of the section of smooth muscle under low power of the microscope, showing the longitudinally cut smooth muscle fibers and transversely cut smooth muscle fibers. This is the photograph taken under high power to show magnified image of the transverse and longitudinal sections. The next 4 slides give the comparison of features between all the 3 muscles. You can pause the video and go through all the features. With this, we complete the histology of smooth muscle. We have discussed light microscopic features, ultra microscopic features, arrangement of fibers, variation of organization of muscle fibers based on function, nerve supply, and features identified under the light microscope with photographs of the sections. In the next session, we will discuss on the clinical anatomy of the muscular tissue.